are the different objectives and questions to keep in mind as we go through this content. And to actually start talking about the brain, what we're going to need to do is actually look at how the embryonic development of the brain actually takes place and the spinal cord. Because how it actually develops will actually define the structural divisions that we see. And you can look at the brain in different ways. You can look at the brain and break it up into structural divisions. You can break it up in functional divisions. We're going to break it up in structural divisions and talk about the function. And to talk about it in terms of the structural divisions, it's really important to look at the development of the brain. So we kind of have to back up and look at embryonic development, and there's this activity called gastrulation. It's Gastrulatin. It's called gastrulation. Gastrulation is going to be occurring during the 3rd week of gestation for the human embryo. And gastrulation is important because it establishes the 3 primary germ layers of ectoderm, endoderm, and mesoderm. And from those 3 primary germ layers, we're going to these primitive tissues, all other body tissues and organs will be derived. So we have to lay down these 3 layers, and then from these 3 layers, we're going to rearrange them and then create the different tissues and organs of the body. So gastrulation, hugely important, happening very early, 3rd week of gestation for the human embryo. This is actually looking at a mouse embryo, gestational day 7, approximately age 17. In the human embryo, the head would be up in this region here, the tail back in that region there. And you can see that through this gastrulation, we're actually setting up the 3 primary germ layers. We're going to have our ectoderm in this position here, our mesoderm, and our endoderm. And as I mentioned, from these 3 primary or primitive germ layers, we're going to get our basically our other structures, our other tissues, other organs of the human body. And so you don't have to memorize this chart. I'm going to give you sort of a way to kind of think about each one of these. First is the ectoderm. Ectoderm from this, think of it as the outer skin. There's 2 things you are responsible for realizing it creates from the ectoderm, and you're going to get the epidermis of the skin, so that's where the outer skin is, but all the nervous tissue, so things like the brain and the spinal cord, are coming from that ectoderm. So think of ectoderm as the outer skin. Well, if we have an outer skin, we also have an inner skin, and our inner skin is going to be coming from our endoderm. The endoderm, from this, we're going to get all the epithelium lining a lot of our passageways, so lining the digestive tract, lining the respiratory tract, lining the reproductive tract, right? Lining the urinary tract. And so think of this as the inner skin. Then you need a filler in between, and that's going to be your mesoderm. And so for mesoderm, we get all the filler. We get things like skeletal muscle, smooth muscle, cardiac muscle, cartilage, bone. So this is your filler material. Well, if we're interested in developing the nervous tissue, specifically the brain and the spinal cord, we're obviously interested in the ectoderm. So we're going to be interested in that ectoderm for this particular unit. We have our 30 primary germ layers, which we set down during gastrulation, and now we're going to rearrange them to form the different body organs and organ systems. The rearranging of these 3 primary germ layers is called organogenesis. Organogenesis. And organogenesis leads to the development of the different body organs and organ systems. And organogenesis occurs from embryonic period from the 3rd to the 8th week of development. And it gives you your main organ systems. The first organogenesis that takes place, because remember it's happening from the 3rd week to the 8th week, it's this rearranging of these different tissues. The first organogenesis that's going to take place is called neurulation. Neurulation, Neurulation. So that's occurring towards the 3rd week of development, ends about day 28. Neurulation is the differentiation of the ectoderm that's going to give you the brain and the spinal cord. So neurulation is going to be using that ectoderm to give you the brain and the spinal cord. So here we see that embryo again, and again, we're very interested in that ectoderm. Remember that ectoderm is this tissue here, which we highlight in the different blue colors. And what's gonna happen to, this is the midline of the embryo, the head is gonna be developing up in this area, tail back in that area. What's gonna be happening to this ectoderm is you can see along the midline, it's starting to thicken up. So you can see it's starting to thicken up. And it forms what's called a neural plate. And so the cells of this neural plate are now called the neuroectoderm. So this is now neuroectoderm. The neural plate begins to invaginate along the midline, forming a groove. This is called the neural groove. So it starts, while the lateral edges start to puff up, forming the neural folds. That neural groove is going to continue to deepen, and we'll look at some cartoon representations showing this, but that neural groove is going to continue to invaginate along the midline, and it's going to cause the folds to come closer and closer together. So these are going to come closer and closer together. At the same time, all this neuroectoderm is going to start to sink down into the mesoderm. So it's coming away from the surface. As those folds come together, it creates a tube with a lumen and tissue around the outside, that neuroectoderm, and that's called the neural tube. So neurulation leads to the formation of this neural tube. Failure to close the neural tube can lead to different disorders. If it doesn't close in the area of where the brain is going to develop, it creates what's called anencephaly. Never heard of that. And if it doesn't close in the area where the spinal cord is going to form, that's going to form spina bifida. So here what they've done is we're looking at the dorsal side of the embryo. Head's going to be up this area, tail down at this area. And what they've done is they've kind of just pulled off that that ectoderm. So here's the ectoderm that we saw earlier. You can see that it stays very thin along the edges. That's going to be what's going to become the epidermis of the skin. It's going to stay at the surface. But along the midline, it thickens up, forming the neural plate. So now this is all neural ectoderm. And we can see that it also starts to puff up on the sides, and those are the neural folds. The neural folds are going to start coming towards each other. This neural groove is going to continue to invaginate. And you can see that now we're going to have these folds come together, and it forms a tube, which is no longer at the surface. At the edges of the neural folds, you can also see that there's these group of cells that leave called the neural crest cells. The neural crest cells are going to go out and form a number of different things. Does anybody remember one of those major items during the hormone lecture in histology? So remember the neural crest cells? These neural crest cells go and end up a lot of different places around the body, creating different things. Things like parts of the heart, cranial facial features. I'll give you a little hint. They also end up in the middle of the adrenal glands, in the adrenal medulla, and form a population of cells that are very important.
Chromaffin?
The chromaffin cells. Because these neural crest cells are going out and also forming a lot of ganglia around the body as well. And they also go form those chromaffin cells, part of the autonomic nervous system. Here's another way to look at this again. So again, they've taken a section through the embryo. They've left the ectoderm by itself. You can see the neural plate developing here. Here's the groove, here are the folds coming towards each other. Here you can see it within the full embryo. So here's your embryo here, and you can see that this is the tissue that we're looking at specifically. That's going to continue to invaginate. We're going to see the folds coming together, and now you have a tube. It's from this neural tube we get the brain and the spinal cord. It's from this neural tube we're going to get the brain and the spinal cord. And when we look at how this actually works, It actually, it's not like the tube forms all at once, the entire tube forms all at once. It actually works like a zipper working in either direction. This is gonna be the head of the embryo up here, tail is back here, and we're gonna see that this is gestational day 8 in a mouse, it's about 23 days in a human, and the formation of that neural tube first starts in the cervical region, and it works like a zipper towards the head and towards the tail, forming that tube. And here you can see the formation of that neural tube. You can see here's that surface ectoderm that was left at the surface. This is all mesoderm. And within that mesoderm, you now see that neural ectoderm, which is forming this tube. Here's the neural ectoderm, and there's the lumen. And it's from this neural tube you're going to get the brain and the spinal cord. The anterior portion of the neural tube is going to give you the brain. The posterior portion is going to give you the spinal cord. So this portion is going to develop into the brain. This portion of the tube is going to develop into the spinal cord. Yeah?
Does the formation of the tube happen simultaneously? Because you said that if it doesn't close in one region or if it closes in another, that causes a different pathology. So
It takes a couple days for it to zip up. So going back to that picture with the zipper. So it's going to happen over a couple days. So first in the cervical region, so it's going to close here first, work its way this direction, and work its way towards the tail. So it's going to be over time that it's going to zip up over a couple days. Okay. Now, let's look at what actually happens to that tube, because remember, it's how this develops that's going to give us the structural organization to the brain. So here's our neural tube. It just looks like a regular straw. But over time, things start to happen. Now, the posterior portion is going to become the spinal cord. The anterior portion is going to go through some changes. So that anterior portion is going to go through some changes. And we're going to see that we're going to have some areas that dilate, some other areas that constrict a little. And it creates what are called the primary brain vesicles. And this is around week 5. And we're going to see that the most anterior portion is called the prosencephalon, also known as the forebrain. We come back a little bit, and over posteriorly, this is going to be the mesencephalon, also known as the midbrain, and then the rhombencephalon, also known as the hindbrain. And that's attached to the rest of the posterior or caudal portion of the neural tube, which will become the spinal cord. A week or so later, we are going to get what are called the secondary brain vesicles. So we get some areas that dilate even more, other areas that constrict a little bit, so we get differential growth, and we're going to see that we have these secondary brain vesicles. From the prosencephalon, we're going to get the telencephalon and the diencephalon. The mesencephalon doesn't change much, so they keep the name mesencephalon. And the rhombencephalon becomes the metencephalon and the myelencephalon. If you're there's a lot of M words here. If you notice, though, they're in alphabetical order from anterior to posterior, so that can help you remember the order of those. It is from these secondary brain vesicles that we get the adult brain structures. So from the telencephalon, we're looking at the tissue of the tube. So if we're thinking of like a straw, we're thinking of the plastic of a straw. From the plastic portion, from those cells, we're going to get the cerebral hemisphere, so the cerebrum. So cortex, white matter, basal nuclei. The lumen is retained as well. And from the lumen, so like the lumen of the straw, we get the lateral ventricles, which contain cerebral spinal fluid. From the diencephalon, we get an area called the diencephalon, which includes the thalamus, hypothalamus, and epithalamus. From the lumen, we get the third ventricle. The mesencephalon, from the tissue of the mesencephalon, we're going to get the midbrain, which is portion of our brainstem. And from the lumen here, we're going to get the cerebral aqueduct. The metencephalon is going to divide a little bit. And from the tissue of the metencephalon, we get our second portion of our brainstem, which is the pons. And hanging off the back of the pons is going to develop the cerebellum. This lumen is retained, and the lumen is going to be part of the fourth ventricle. And from the myelencephalon, from the tissue of the myelencephalon, we're going to get the last portion of our brainstem, which is the medulla oblongata. And from the lumen, the rest of the fourth ventricle. And as I said, The posterior portion of the neural tubes will become the spinal cord. So from the tissue of the posterior portion or caudal portion of the neural tube, you get the tissue of the spinal cord, and the lumen is retained as well, and the lumen is the central canal of the spinal cord. So this explains why the spinal cord is attached to the medulla oblongata. This also explains how we're going to see the flow of cerebral spinal fluid, because these are all connected to one another. Question?
When you say ventricle,
It's a space. So don't think that so ventricle like of the heart, it's a space. So ventricles within the head are going to be spaces that contain cerebral spinal fluid, which is a product from the blood, but it's not blood. But there is going to be a flow associated with it, and we'll talk about that tomorrow. Questions? Yeah.
so, for example, sometimes like, when you're just sitting and your nose just start running, is there a specific ventricle that comes from?
The nose running is probably going to be more from associated with either the mucous membranes of the nose or from the eye, because we have that nasolacrimal duct coming down. There has been, and I have heard of stories where somebody has had some sort of brain injury, traumatic brain injury, and they have a crack in the base of the skull, and they actually can push cerebral spinal fluid out of their nose. That's a bad thing. So they can do certain tests to make sure it's to see if it's CSF, but I have heard of that before. So you should not have CSF coming out your nose. If you didn't like that chart, this does the same thing. So you can see here's our primary brain vesicles, here's our secondary brain vesicles, here's the walls, what the adult structures of the walls are, and here's what the spaces become. And what we're going to do is we're going to walk our way through the brain, through the systematic structural organization. So we're going to start with the cerebral hemispheres and with the medulla and the cerebellum. So this just shows you that organization and how, and so when we look, this is going to be an embryo, mouse embryo. This is going to be gestational day 10 for the mouse, approximately 5 weeks for the human. This is going to be the area of the head. This is all head here. And this is going to be face here. This is actually the heart tube right here, sitting, so your heart actually sits out in front of your face for a while during development. And so we saw that sagittal section come through, and it's cutting through that neural tube. And so you can see the primary brain vesicles. You can see the forebrain, the midbrain, and the hindbrain. And from those, then we get the secondary brain vesicles. So from the telencephalon, we're going to get the cerebral hemispheres. From the diencephalon, we get the diencephalon, which is a lot of thalamus, also hypothalamus and epithalamus. Midbrain does not divide. And then we see the hindbrain dividing into the metencephalon and the myelencephalon. And you can see the different divisions there. And as this is happening, the neural tube is going through a lot of growth during this time. And so we can see that at 5 weeks, we start getting these, here's where the spinal cord is. This is the anterior portion of the neural tube here. You can see it's going through some areas that are getting larger, increasing in size, and other areas that are going to constrict a little bit. We also have flexures that come into play, so areas where we have some bends come into play. But the interesting thing is not all parts of the brain are going to develop at the same rate. What is growing incredibly rapidly are going to be the cells of the telencephalon. So what's going to become the cerebral hemispheres. And so, but if you think about it, the skull's not growing very quickly, but the telencephalon is. So this telencephalon is like, I want to grow, I want to get all this space, I want to have all this surface area for all these neurons. But we cannot grow very quickly within a confined space, so it needs to figure out a way to grow and create a lot of surface area. So first it's going to take on this interesting shape where it actually grows posteriorly and laterally and starts to actually envelop the diencephalon. So if you want to look for things like the thalamus, the hypothalamus, the epithalamus, they're deep within the brain, and it's because of that rapid growth of the telencephalon around those areas. Well, those neurons want to have a lot of surface areas, and so because we can only go so big, we're actually going to start to create sulci, and fissures. And so we're gonna have a lot of different developments of these different folds associated with the cerebellum, with the cerebrum, as well as the cerebellum, because we wanna have a lot of surface area associated with these neurons. It's all because the telencephalon wants to grow very quickly, it wants to have a lot of surface area, but it can't grow very quickly because the skull is, it can't grow large because of the fact that we have the skull developing at a slower rate. But all of these folds, as we know, are gonna be important to create a lot of surface area, and we learned that hopefully from histology. Folds are very important for creating surface area. So some important terminology, we mentioned some of these yesterday, but I wanna make sure everybody's on the same page when we talk about these. We're gonna see that in the central nervous system, when you look at the brain, even without it stained, you're gonna see that there's white matter and gray matter. White matter is mostly composed of myelinated axons, and it's white because of the myelin. Remember that myelin is just, and this is central nervous system. Anybody remember what cells produce myelin in the central nervous system?
Oligodendrocytes.
Oligodendrocytes, good. So those oligodendrocytes are sending out those processes and wrapping around. Well, that's plasma membrane, which are phospholipids, which are fats, which is white. And so that gets that tissue, that white appearance associated with it. We also have gray matter, which is composed of a lot of other stuff. Dendrites, unmyelinated axons, axon terminals. But also that gray matter has a lot of neuronal cell bodies. So very important because the neuronal cell bodies are there, which is the biosynthetic center for the neuron. If it's the biosynthetic center, it's going to have a lot of rough endoplasmic reticulum, which we know are called the Nissl bodies. And that sort of gives it that gray appearance also because of the absence of the myelin. So we saw that the brain, the telencephalon, took on that very rapid growth. And so we see that associated with the cerebrum, we're going to have these folds associated with it. The gyri, gyrus is singular, gyri is plural. The gyrus is going to be an elevated fold within the brain. So it's going to be an elevated fold of the brain. And as you go up that fold and down, the shallow grooves between the folds are called the sulci. Sulci is plural, sulcus is singular. There's also going to be some deep indentations, and those are known as fissures. So if it's very shallow, it's a sulcus. If it's very deep, it's going to be a fissure. And a lot of those fissures are utilized to separate different parts of the cerebrum. We'll also see that the cerebellum also has folds associated with it as well, but we call them a little bit different names. Remember that clusters of neuronal cell bodies in the peripheral nervous system is called a ganglion. In the central nervous system, it's called a nucleus. The nucleus will be part of that gray matter. And we're going to see bundles of neuronal axons also in the central nervous system. They create what are called tracts. If it's in the peripheral nervous system, we know it's a nerve. So if we remove the calvaria and look into the cranium, we're going to see 3 levels or fossa, or fossae. Each is a stair-step down pattern as you go from anterior to posterior, and we looked at this yesterday. So you can see this is the anterior cranial fossa. You come over the lesser wings of the sphenoid bone, and you get into the middle cranial fossa. You then go over the petrous portion of the temporal bone.